Good morning. Welcome to Grand Rounds. It's my great pleasure this morning to introduce our speaker, Dr. Mary Lewinski. Dr. Lewinski is an associate professor of medicine and a staff physician at the VA in the Division of Infectious Diseases and Global Public Health. Dr. Lewinski received her PhD in biomedical sciences and her MD degree from the University of California, San Diego. She then completed her internship and residency as a member of the Physician Scientist Training Pathway, also here at UCSD, followed by an infectious diseases fellowship. Dr. Lewinski is the recipient of an NIH K08 studying HIV-1 modulation of the ubiquitinome and its effects on cell-to-cell transmission. She also serves as the Associate Program Director for the UCSD Infectious Diseases Fellowship, as the Director of the San Diego Center for AIDS Research Developmental Corps, the Assistant Director for the UCSD Medical Scientist Training Program, and the Associate Director for the Physician Scientist Training Pathway in the Department of Medicine here at UCSD. She is a very well-recognized investigator and physician scientist. We're really fortunate to have her here this morning to share her presentation on host defenses and viral countermeasures. Thank you so much, Dr. Lewinski.
Hello, good morning. I hope you're all awake. So as was stated in that very kind introduction, thank you for that. My name is Mary Lewinski. My pronouns are she, her, hers. And being at UCSD, I would like to respectfully acknowledge that I live and work on the land of the Kumeyaay Nation. I have no disclosures to report for this presentation. And before I get started, I was asked to give a little a brief discussion of, kind of inspirations in medicine, things that may be unrelated to what we're talking about today. And I think, while I remain mystified why I had this invitation, given the caliber of our first two speakers, I was very much inspired by their calls to eliminate biases in our practice of medicine. And I think a lot of us went into medicine to provide care to the underserved and reach out to the marginalized. And I think a community that has been long maligned by and neglected by the medical community are trans and non-binary patients. And so I wanted to highlight some work just briefly that Jill Blumenthal and my other colleagues here at UC San Diego have been doing to bring gender affirming care, or to expand gender affirming care here at UC San Diego Health, and also to educate all of us. And I would like to highlight their December 11th virtual meeting for CME credit and invite you all to participate if you're available. I think that transgender, non-binary care, gender affirming care is something that all primary care providers really should be able to provide competently. And, I didn't really encounter this type of training until I was a fellow. Now we're getting better at training medical students in this regard, but I think a lot of us that have been practicing for a while could use some extra education discussion in this regard. So anyway, a shout out to the great work that they're doing, and I hope you can join us for that. And so, as they say, now for something completely different. Today, I'm going to be talking about some mostly basic science work that I've been doing, but also give you some context with some nice work that many other investigators have been doing, looking at viruses and host restriction factors. I will specifically be focusing on the two viruses that take up most of my time, which is HIV and SARS-CoV-2 as of late. So as far as we have experienced in recent years and recent decades, we have found that zoonotic disease is really has had a terrible impact on human health. A lot of, and this is just highlighting some of the viruses that have been important to our health. So HIV, of course, had a non-human primate origin. Ebola virus, thought to have a bat origin, as well as the original SARS coronavirus. Avian influenza virus caused a lot of deaths, has a bird origin. And then SARS-CoV-2, while the reservoir hasn't fully been described, at least some precursors are from bat and maybe an intermediate such as pangolins. So in order for a virus to leap between species or to humans, there needs to be, of course, human exposure to the reservoir and or an intermediate host. And the virus has to be capable of infecting humans. So that means it has to have compatible host factors, which either means that there are shared features, so the virus and the animal host uses the same receptor or a very analogous receptor in the human host, or that virus has mutated to be able to use the receptor and such in the human host. And also, these viruses need to be able to counteract host defenses. So, there are a lot of barriers to spillover. And when the holes in the Swiss cheese line up, that's when we get this spillover event from animals to humans. And there are a lot of different disciplines that really look into a lot of these areas, such as the reservoir, what's going on with the animal ecology, population biology of the animal reservoir, and their density and distribution, and then the prevalence of the pathogen in that reservoir and the intensity of the infection in that, how likely the pathogen is to be released from the reservoir host, how sick it makes them, if the pathogen can survive in the environment, how easily it spreads away from its reservoir host, and then how much humans are being exposed to these reservoirs, based on our encroachment on their ecosystem and our behavior. And then, the area that I have focused my work on, of course, is the kind of microbiology, innate adaptive immunity, molecular virology of all of it. And, there are a number of barriers to overcome for the virus in this case. In this regard, the physical barriers, right? It has to kind of get into the body somehow through the skin, mucous membranes. We have a lot of physical barriers set up to protect us from pathogens, stomach acid, that sort of thing. And of course, the virus would have to be able to use the human equivalent of their receptor. And then we have, of course, interferon-induced and other innate immune responses. And these can be triggered very soon after after we recognize that we have encountered this pathogen. And this can result in apoptosis or interferon-induced resistance that prevents the organism from really taking hold. And then, of course, there are interfering defensive proteins in the host cell. So specific proteins that block the replication of the virus if it were to make it into the cell. And then viruses, of course, need the cellular machinery to some extent to replicate. And so there need to be particular host factors, and they may be specific to the reservoir host and not humans. And so the virus either needs to be able to mutate, there needs to be enough diversity in the infecting virus that it can adapt to use the host factors, or it will not succeed. And so, there's this evolutionary arms race throughout history where, we have the host defenses and we have the viruses trying to counteract them. And so where viral factors are able, so in this case, so this is a nice figure out of a review from the Emmerman lab a few years ago. You have a viral antagonist that is able to bind to, and maybe in some cases degrade or otherwise inactivate, the host restriction factor, that is the host defense against one aspect of viral replication. And this enables the virus to replicate. And so, of course, there's a fitness cost if that viral replication is in any way toxic to the human host. There's a fitness cost to allowing that virus to replicate. And so over time, of course, humans evolve a lot more slowly than viruses, but over time, there may be escape mutations in the host that enable restriction. So, in this case, this blue host restriction factor may evolve the binding site for the viral antagonist. And so in that case, it may still be able to restrict viral replication because the viral antagonist is not able to counteract it. And so then this kind of moves on. Then that's a selective pressure now on the virus to evolve an antagonist that can now bind to and disable or otherwise get around this host variant, and so on. And so there's this arms race that goes on and on through the millennia. And these historical conflicts between the virus and the host actually leave marks in our human genome. And so a lot of groups like the Emmerman group kind of look at the sequences and the sequence diversity of a lot of these host restriction factors and such, and cofactors. And basically in the absence of genetic conflict, genes evolve under negative or purifying selection, which means there's a lower rate of non-synonymous mutations than of synonymous mutations, which means that the dn over ds is less than 1, and that's what would happen kind of here. But if there is a genetic conflict from a viral antagonist, then for instance, the binding site for that antagonist on the host restriction factor may evolve away such that the viral antagonist can no longer bind. So the host gene in this case will accumulate non-synonymous mutations. So the dn over ds would be greater than 1. And so now I'm going to pivot a bit to HIV accessory proteins, and some of this will illustrate what I've been talking about. I also would like to highlight the amazing art, if you haven't seen it, of David Goodsell, who's a scientist at Scripps, who does these paintings that are to scale of viruses and other pathogens, and they're freely available online if you ever have occasion to need something like that. So the origins of HIV, some of you may be familiar with this, some maybe not, but HIV comes from simian immunodeficiency viruses, which are circulating naturally found in African monkeys and apes. And in general, they're not pathogenic in their natural host. So the sort of arms race between the human factors and the viral factors have reached a sort of detente where, the virus gets to spread in the population, but doesn't cause enough harm to the host for there to be an intense selective pressure that would eliminate it. And so, that's all fine and good. But then if the virus is able to leap across to another species that is not as well adapted, then it can cause disease. And that's in fact what happened with HIV. So there's HIV-2, which is a result or a number of different strains from different cross-species transmissions of a City-Mangabe SIV into humans. And then pandemic HIV-1, which we normally think about when we think about HIV, is from a chimpanzee SIV. And sort of interestingly, this pandemic HIV-1 group M from this chimpanzee SIV is actually pathogenic in chimps. So that goes against what I just told you about these usual circulating monkey SIVs. And that is because SIV chimpanzee is itself a recombinant virus between a couple of different SIVs. And so it ended up being pathogenic in the host where it was established. And there was nice work done about a decade ago by Beatrice Hahn and her group that actually looked at stool from different groups of chimpanzees across Africa, and found that the closest relative to group M HIV was actually from a specific group of chimpanzees in southeastern Cameroon. And they suspect, based on sequence analysis from early human bank tissue samples and evolutionary analysis, that the initial transmissions probably occurred into the human population. The spillover events occurred through bushmeat hunting and exposure to infected blood. And then really basically this area where the chimps live is upstream from Kinshasa, which at the time was Leopoldville on the Congo River in the early 20th century, where there was a lot of disruption of traditional social structure, a lot of colonialism. So this enabled the spread in the human population and more adaptation to the human population. So this is the HIV genome, about a third the size of the SARS-CoV-2 genome, which I'll discuss a little later. But there are these 4 so-called accessory genes in the HIV genome, and they're called accessory genes because they're not strictly required for replication, although it won't be very efficient. In certain cell culture systems, you can get the virus to replicate without these. And they have a number of different functions. In fact, just about they're multifunctional in ways and in clever ways, just kind of showing how efficient viruses can be when they evolve. So NEF, for instance, here as an early gene that downregulates CD4 and MHC class 1 from the cell surface. It enhances virion infectivity through a number of actions. VIF here counteracts a host cell restriction factor called ApoBac3G, which causes hypermutation of the viral DNA if it gets incorporated into virions. There's VPR, which is still somewhat mysterious, but it is known to aid in the infection of non-dividing cells. And then there's VPU, which counteracts a host cell protein I'll spend a bit of time on called BST2, also somewhat renamed tetherin, which traps budding virions at the cell membrane. And VPU has other effects on other cellular proteins as well. So host restriction factors attack many stages of the viral replication cycle, and they may be constitutively expressed as part of intrinsic immunity, or they can be induced, often by interferons like BST2 tetherin. And the viral antagonists, on the other hand, they're often these accessory proteins, but not always. And the counteraction may require co-opting of cellular cofactors, and I'll talk about that. And they can often be the key to cross-species transmission in the host range, because if a virus does not encode an antagonist that can interact appropriately with the restriction factor, then it won't be able to replicate very well in the host. And so, we talk about, so as far as host cell fusion, there are these proteins, this membrane proteins called syrinx, as well as IFID-M, which block various aspects of fusion of the virion. And then there are cytoplasmic factors like TRIM5 alpha that block uncoating. There are factors such as SAMHD1 and ApoBac3G, which affect the reverse transcription process and the availability of DNTPs for that process. There's MX2, which can affect nuclear import of the pre-integration complex. And then with assembly and budding, there are proteins like BST2, which basically trap the virus on the cell surface. And then in green here are various areas where these viral antagonists, these accessory proteins for HIV, interact with these host restriction factors to disable their activity. And so one of the clever things that HIV, 3 of the HIV accessory proteins do is they act as substrate adapters for the ubiquitin proteasome system. So they bind and recruit these systems to their cellular restriction factor target, like VIF and ApoBac3G, or VPU and BST2, VPR, and a few different things, including UNG2. And they basically trick the cell into ubiquitinating and degrading their own protective proteins. And so, you could think of ways that this sort of thing could be adapted, even therapeutically. But it's a very clever way that the viruses have evolved to really use the cell's own machinery against its protection. And so BST2 is this is also called tetherin, is a pretty interesting protein. It is interferon induced, although a lot of cell lines do have some constitutive expression as well. It has this odd structure where there's a cytoplasmic N-terminus, a transmembrane domain, and then a coiled extracellular domain that can interact with other BST2 proteins, and then a C-terminal GPI anchor. So it's basically anchored in 2 membranes. And this is how it can kind of hold on to budding virions and keep them trapped at the cell surface. And in general, BST2 traffics through the ER and Golgi. This is how it kind of starts to interact with some of the viral proteins, and then ultimately localizes to both the plasma membrane and in endosomes. And so this is really important in restricting certain enveloped viruses. So all the HIV, SIVs, all the retroviruses that have been tested are restricted to some degree. Viruses like Ebola, Marburg, Lassa, VSV, Nipah, and then of course the SARS coronaviruses also have been found to be restricted by BST2. And there are various viral countermeasures, various strategies for antagonizing this restriction factor. So what the first to really be recognized as KSHV actually encodes a ubiquitin ligase that directly degrades it. And then SIV encodes NEF. HIV, of course, HIV1, VPU, but HIV2, as well as FIV and Ebola, use their envelope glycoprotein. And in some cases, this leads to the degradation of BST2. In some cases, it leads just to the downregulation. In some cases, it leads just to pushing it away from sites of viral assembly or some combination of that. Interestingly, there is evidence of this species specificity in BST2 antagonism. So for instance, most SIVs use NEF against BST2, and NEF interacts with this 5 amino acid stretch that are in a lot of these monkey BST2s. But interestingly, HIV1 and the precursor use their VPU instead of NEF. And part of the reason for that is the human BST2 actually has deleted these 5 amino acids that interact with the NEFs of SIVs. So this sort of suggests that our genome was in some way marked by some ancient interaction where the BST2 is being counteracted through this stretch of amino acids, and there was some selection there. Interestingly, HIV2, because the precursor SIV does not encode a VPU, had to evolve to use its envelope to counteract BST2. So you can see it in the mark of these genomes, and also this underscores how important BST2 really is, and the counteraction of that really is for viral replication. And so VPU is, I kind of alluded to this, is a very multifunctional protein. It's not a very big protein, but it acts against BST2 in 2 different ways, and I'll talk about that shortly. But it both downregulates it from the cell surface, which is the site of HIV assembly, and then it also excludes BST2 in the plane of the plasma membrane from assembly sites. And we'll talk about a little bit about what we know about how it does that. It also VPU targets CD4 for degradation, which is interesting. CD4 is the receptor for HIV. And it's interesting that the virus devotes, or basically has 3 proteins that have anti-CD4 activity. So once the cell is infected, it does a lot to try to downregulate CD4. And there's good reason immunologically why the virus may want to do this. So when envelope binds to CD4, there's a conformational change that where they're called so-called CD4 induced epitopes, which are exposed. And this really sensitizes the virions where there's this triggered envelope to antibody-dependent cell-mediated cytotoxicity. And in fact, if you delete NEF and VPU from the virus, you can enhance ADCC by exposing these epitopes with the CD4 envelope interaction. VPU also downregulates NTBA, a natural killer cell co-activating ligand, and CD1D, which is an MHC-like lipid antigen presenting molecule. And the consequences of these activities aren't fully understood, but it seems like immune modulation is a likely result. And just interestingly, only the group M pandemic HIV-1 VPU has both anti-BST2 and anti-CD4 activity. So this may be really forecasting why this particular HIV went worldwide like it did. So the key structural features of VPU, it has this transmembrane domain, it has cytoplasmic domain, and the cytoplasmic domain has these alpha helices. It's not totally clear how they interact with the membrane, but they're one or more of them may at different points in time orient themselves such that they interact with the membrane or other cellular factors. And there are trafficking motifs. There are also these phosphoserines that are recognized by beta TRCP, and that's important in some of the degradative activities of VPU. And then there are some membrane interacting amino acids in the C-terminus, which I will talk about a bit. And so then there are different clades of HIV, of course, and so this is just a few of them showing you how conserved some of these trafficking motifs and such are. And so VPU interacts with BST2 through its transmembrane domain, and this is sort of this is a look at that. And it targets BST2 for degradation by using these phosphoserines, these very conserved serine 52 and 56, which, when phosphorylated, bind and recruit beta TRCP and its ubiquitin ligase complex that targets BST2 for degradation. It also can downregulate BST2 from the cell surface through misdirection of endosomal trafficking. So BST2 is kind of recycled from the plasma membrane, and it can get locked in the pathway by its interaction with VPU. And then more recently described was this idea that VPU can direct BST2 away from sites of viral assembly on the plasma membrane. And it's really this activity that best correlates with VPU's ability to enhance the virus release. And so one of the questions that we asked was, if BST2 is so important for cross-species transmission, as I mentioned, could it impact human-to-human transmission? And so there are these viruses that have been described called so-called transmitted founder viruses. And we hypothesize that maybe as a group, they have superior anti-BST2 activity compared to chronic isolates. And why we might believe that is, these transmitted founder viruses, in order to establish infection in their new host, had to be very efficient at doing that. They had to do that in the face of a big interferon response in the host, which would have upregulated BST2 in the target cells. And they basically, these transmitted founders have kind of won the battle through the bottleneck. So maybe there's something special about them in this way. And so so these transmitted founder viruses are the kind of represent the very few or one virus that kind of makes it through the bottleneck and then evolves in the host into a diverse quasi-species that is under selection by the immune response. And Beatrice Hahn, Shaw, and colleagues basically constructed years ago panels of these transmitted founder infectious molecular clones. And they did this using single genome amplification and in phylogenetic inference, and basically constructed what they believed to be what must have been representative of the transmitted virus in the host. And it has been described that these viruses as a group are better able to replicate in the presence of type 1 interferons. So suggesting what I had said earlier, that early in infection, the successful viruses have to be able to replicate in that setting. And so we set up a system to test the activity of these VPUs, or the primary two activities of these VPUs, that is, CD4 downregulation and BST2 downregulation from the cell surface. And in this case, we're really just looking at how in the cells that have been transfected, which is marked by the GFP transfection marker, compared to a well-characterized laboratory strain of HIV-1, the NL43, we looked at some of these transmitted founders. And if you look at them as a group, this is compared to the NL43, you do get a little bit more CD4 downregulation in some of these, but not a whole lot more. As far as BST2 downregulation, there really wasn't, as a group, a whole lot of difference. So, we thought, okay, that doesn't really confirm our hypothesis at all. If you look at virion release, that is, you look in the supernatant, how much virus is actually getting out of the cell. There wasn't, as a group, the transmitted founder VPUs didn't act in a particularly elevated way. However, we did notice there was one particular VPU that was particularly bad at enhancing virion release. So we kept an eye on that, and to sort of examine this further using the primary infection samples from the primary infection cohort that managed here at UCSD by Susan Little, we looked at some acute and chronic VPUs, that is a virus that was isolated early in infection and then later, and we found this sort of interesting pair that where the acute VPU, the isolate from the acute infection was okay at enhancing virion release. The chronic isolate really wasn't. And interestingly enough, both this transmitted founder VPU and this chronic isolate had the same polymorphism, and that was there is this otherwise clade B conserved tryptophan that had been changed to glycine. And so we did some site-directed mutagenesis, a W76G mutagenesis. So we took a couple of good, well-performing VPUs, the NL43, and then one of the transmitted founders that performed well in our assays. And if you changed this a W to a G, you didn't see any difference in BST2 downregulation from the cell surface, but what you did see was a big effect on virion release. And this was basically telling us there was another way in which the VPUs were enhancing virion release that didn't have to do with downregulation from the cell surface. And if you did the reciprocal, where you took these VPUs that weren't so good, and we changed the residue 76 back to a W, again, it didn't affect downregulation from the cell surface, but it did very much enhance virion release. So we knew that this was key, at least in clade B, to this newly described phenotype. And so we were trying to look into the mechanism. This is in the setting of virus infection. I told you about those really important serines, 52, 56. They're really important for directing the degradation of BST2. They're also important for getting it off of the cell surface. So if you mutate those, you see you don't get any downregulation of BST2. You get poor virion release enhancement here. The W76G mutation did just fine at downregulating from the cell surface, very similar to wild type, although it had some trouble getting up to the level of virion release enhancement, as we just discussed. And then if you mutated the serines as well as the W, basically you completely abrogate VPU's ability to release, to enhance virion release. And so this combination of both degrading, moving BST2 off the cell surface that's mediated by these serines, and whatever this W is doing to enhance virion release is really what was key to this activity. And so we looked at what was called, what we and others call kind of displacement of BST2. So that is, so the total level of BST2 on the cell surface that we measure by flow cytometry wasn't decreasing, but there was a way to really show that BST2 was being pushed away from sites of viral assembly, which was happening in the plasma membrane, and that this is dependent on this W76. And so here we're just kind of looking at overlap. If you look at when there is no VPU, you get a lot of overlap between envelope and BST2. When you have wild type around, you don't. And then with the W76G, again, you get a lot of yellow showing that it's not able to push BST2 away from the sites of assembly as shown by the viral envelope. And so we had looked, although I'm not showing the data here, we looked at the ability of VPU to reorganize lipid rafts or tetraspanin enriched domains, and that didn't appear to be something that was being reorganized. So we think there's some direct effect of the W76 on trafficking or reorganization of BST2 in the membrane. And we worked with the Pella lab in the Department of Chemistry here that were able to model this C-terminal domain of BST2 or VPU, and they show that the W76 really kind of sticks out. So it's reasonable to assume that this would maybe be sticking into the membrane and perhaps reorienting the cytoplasmic domain such that it was interacting with host factors, different host factors, and basically moving BST2 around in the plane of the membrane. And so that is still something. These are still mechanistic studies that are ongoing. In the ensuing years, there are a number of different mostly membrane proteins that have been shown to be reorganized by VPU in one way or another, either degraded or pushed away or sequestered in endosomal compartments. And so a lot of this remains under study, and we don't fully understand a lot of the implications of this, but it's clear that the virus has made a great use of this small so-called accessory protein to really reprogram cellular pathways to its benefit. And so now I'll move on a little to SARS-CoV-2 with my closing time here. So of course, a lot of us, I think, that did basic virology research and a lot of immunology pivoted a bit and kind of applied what we knew of virology in our systems to urgently studying SARS-CoV-2. And so the coronavirus replication cycle is quite a bit different from the HIV cycle. You get endocyte, often the spike protein binds to the ACE2 receptor. Depending on the cell type, you get endocytosis and the viral protein particles escape the endosome. And then you get transcription and translation at the ER membrane to get the accessory and the structural proteins. So SARS-CoV-2 has a number of accessory proteins, of course. And then the S, E, and M proteins get inserted into the ER, and through the secretory pathway, they actually assemble not near the plasma membrane like HIV, but in this ER Golgi intermediate compartment, or ERGIC. And the virions bud from the ERGIC and traffic in vesicles through the trans-Golgi network to be released by exocytosis. So we, in collaboration with the Shanda Lab, which at the time was at Stanford Burnham, but is now at Scripps, and Laura Martin Sancho, they conducted a screen for interferon stimulated genes that restrict SARS-CoV-2 replication. And they found, of course, that SARS-CoV-2 is sensitive to type 1 interferons, and 65 of the 399 interferon stimulated genes that they tested did restrict SARS-CoV-2. And most of the ISGs that were restrictive reside in the ER, the Golgi. So, or they regulate ERAD, lipid metabolism, lipid membrane composition, and vesicle transport. And all that makes sense based on what I told you about the replication cycle of SARS-CoV-2. And sure enough, BST2, when overexpressed, was shown to inhibit viral infection. And this here is in two different cell lines. And so, we undertook studies to kind of figure out what exactly was going on here, because as I described to you, BST2, how it acts against HIV or is thought to act against HIV and other enveloped viruses is it tethers them to the plasma membrane. And if SARS-CoV-2 isn't replicating at the plasma membrane, then what exactly is BST2 doing to restrict it? What we do know, though, is if you knock down BST2, you get more viral spread. So this is in a knockout HeLa line. You get a higher level of viral replication and spread in the culture, in the knockout culture. And then in CALU3s, similarly, if you knock it down, you get more spread. And then RF9 is just another restriction factor that you see. Its knockdown also allows for additional spread. And we screened some of the potential antagonists of and accessory proteins of SARS-CoV-2 for anti-BSD2 activity. And it turns out ORF7A appears to antagonize activity such that when you have BSD2 expressed in cells, you get a difference in spread whether or not ORF7A is absent here in the dashed line or present. Whereas in the absence of BSD2, there isn't a difference in spread in this system. And so what is ORF7A? It is an accessory protein that is packaged into virions. It's actually pretty similar to the SARS-1, the original SARS coronavirus ORF7A. It's a type 1 transmembrane protein that has an N-terminal signal peptide, and it has these beta strands in the extracellular domain have an IG fold. And while sequence-wise, it is not really similar to the D1 of ICAM1 and 2, structure-wise, it is. So, so there has been some studies on the SARS-1 side about potential analogous function and binding to ICAM1, for instance. And then it has a stalk, a transmembrane domain, and an intracellular C-terminal tail that has an ER retrieval motif. And as I mentioned, SARS-CoV-1, which I have here above, and then the second line is the SARS-CoV-2 ORF7A. They're very similar. That I've highlighted in red the residues that differ. But SARS-CoV-1 studies, of course, were years ahead of what we've been able to do so far. But it was found to, for instance, induce apoptosis via P38 MAPK activation. It binds, as you might expect from its structural similarity to ICAM1, to the alpha L subunit of LFA1 on lymphocytes. So it's not really clear what exactly that is doing. It was just shown in the early studies of SARS-1 to bind without really a discussion of potential consequences, but one might imagine that could have effects on the immune response. And then it was shown to block the restrictive activity of BSD2. The mechanism of this effect for SARS-1 or ORF7A is not really clear. There was a study that posited basically it had to do with ORF7A preventing the glycosylation of BSD2. So BSD2 is a heavily glycosylated protein, but for reasons I'll show you, that mechanism, at least for SARS-CoV-2 7A, doesn't appear to be what's going on. So we found that using HIV-1 VPU as a control, of course, because VPU is our kind of model BSD2 antagonist. ORF7A, we found this is provided in trans in a VLP, a virus-like particle system, does get packaged into virions. So this is the VLP set here. You can see ORF7A in the virions. And it does counteract BSD2 to allow for a lot more virion release. So compared to basically here where you have BSD2 but no antagonist, you get a lot more release of the nucleocapsid protein in the supernatant. And this is the similar findings with an ELISA for the supernatant instead of the Western blot, but you see both the addition of ORF7A or VPU enhances the release of the SARS-2 N protein in the supernatant. And we did show that BSD2 co-immunoprecipitates with ORF7A. So this is just a lysate. These are the IPs. So ORF7A is here. It's bringing down. So this is ORF7A here, the flag tag ORF7A, and it's bringing down BSD2. This is VPU as a control, is even a lot more highly expressed and pulls it down. And then if you look at glycosylation mutant of BSD2, the interaction still stands. So, and then if you look at just the glycosylation in the cell lysates, there doesn't appear to be a difference in the size of the BSD2, for instance, when ORF7A is expressed. But unlike VPU, ORF7A does not appear to downregulate the cell surface BST2. And from what I told you about where the SARS-CoV-2 assembles, maybe this shouldn't be too much of a surprise. But so here, basically, as a transfection marker, we have GFP. We have the different components of our VLPs as well as BST2, and you can see whereas VPU downregulates the surface BST2, or if 7A pretty much is like as if there's no antagonist present. And then this is just the MFIs to illustrate that. So that does not appear to be the mechanism. And so we're still working on what the mechanism could be. The ORF7A, based on what I told you about how SARS-1 ORF7A interacts with LFA1, perhaps there's a role in sensing or signaling that could be affecting either the pathogenesis of SARS-CoV-2 or its ability to just general ability to replicate. And then, what is the clinical relevance of this activity? There have been ORF7A mutants described in the population, or rather sequenced in the population. They haven't really been connected to a phenotype. And then, could there be clues to the reservoir of SARS-CoV-2, since that hasn't been fully identified, or maybe things that tell us about spillover into the human population? So this is a bioarchives look at different bat isoforms of BST2, also called tetherin. And, bats, actually, there's been a lot of selection in bat BST2. And considering how bats harbor a lot of different viruses that have kind of spilled over into the human population and caused some trouble, including Ebola and then the coronaviruses. There's likely a lot to be learned here in looking at how the different BST2s interact with SARS coronaviruses. And so I would just like to acknowledge, I've had one of the reasons I've stuck around UCSD so long is because of so many amazing mentors and collaborators over the years, and this work really wouldn't be possible without them. So John Guatelli has been a longtime postdoctoral mentor as well as mentor on my K. And his lab, it's talented people in his lab, including Charlotte Stoneham. And then a lot of the VPU work was done with a master's student, Moeen Jafari, who's now a physician. Sumit Chanda lab, which was at Sanford Burnham, recently moved to Scripps, and his talented scientists that work with him, including Laura Martin Sancho. The Stan Appella lab, a longtime collaborator of the Guatelli group in the Department of Chemistry and Biochemistry. And then Bruce Torbett lab, previously at Scripps, now at the University of Washington. And then my funding sources. Thank you all for your attention. I believe we have a few minutes for questions, and I will see if I am able to look at this. Okay. So.
Thank you so much for that really impressive.
Oh, sorry, you're very quiet. It may be me.
I apologize. Thank you, Dr. Lewinski, for that outstanding presentation. That is really an impressive amount of work. We have a question from one of our chief residents, Dr. Jiang. Thank you for this fascinating discussion of innate antiviral immunity. Given the role VPU plays in HIV-1 egress from infected host cells, how might we leverage our understanding of BST2 function to develop new antiretroviral therapies? Thank you, Dr. Jiang.
That's a great question. So, as is obvious, I think from what we discussed today, BST2 is really, really important for envelope virus replication. And so enhancing its activity is definitely a focus of a lot of study. So either I told you it was interferon inducible, so with some caveats, enhancing its expression and in that way can be helpful, at least at certain phases of viral replication. And the other thing I mentioned was ADCC, so that antibody-dependent cytotoxic killing of cells. That's enhanced when viruses are stuck to the cell surface of an infected cell. And so enhancing BST2 activity by counteracting these viral countermeasures really can enhance clearance potentially of the infected cells by enhancing ADCC. And so, you know, in a lot of ways, looking for small molecules or peptides that can block the interaction between these viral countermeasures and BST2, ways for enhancing BST2 expression. So those are the kind of strategies that are ongoing. But it's certainly, unfortunately, there are so many different ways that a lot of these viral antagonists get at BST2. So, you know, it may be playing whack-a-mole a little bit, or have to make a virus-specific inhibitor. For instance, it's unlikely that you're going to find something that prevents all counteraction of BST2, because there are so many ways that these viruses attack it. But yeah, that's certainly a way that we could clear more virus and affect its spread, for sure.
Thank you, Dr. Lewinski. Any other questions?
Well, feel free to email me if you have other questions or you're interested in discussing further. I also want one last plug for that Transgender Healthcare Symposium on December 11th. If you want that information, again, please reach out. I'm happy to disseminate further. Let's see, was there another question? No, I think we're good. Okay. All right. Well, thank you so much.
Thank you, Dr. Lewinski.
have a great
Margaret
day.
has posted the link to the